Program into the speaker. So we have our third day of online webinar instance is production processing and market and development. So there are 300 participants of our Central Agriculture University students and research scientists and faculties and entrepreneur seekers. They have all registered for these programs, and most of them are students. And there are both other students from all over India. They have registered. And today is the third day. So dear participant, so today we have with us a guest speaker, Dr. Pritham Y. Shinte. So he's assistant professor, botany, College of Agriculture, Pune, Maharashtra. So sir will be delivering on the topic seed production, quality control, and value addition. So a little bit brief about his biodata. So he has completed his BSc Agriculture in the year 1992, and then MSc in seed technology in the year 1995 from MPKV Ravuri, and he has completed PhD Seed Science and Technology from Indian Agriculture Research Institute, New Delhi. So he has achievement. He had a teaching experience of 10 years and teaching to the undergraduate. And he has taught a number of courses to the students. And also he has worked in ASRP in maize and wheat. And he is also involved in 8 varieties. He was associated in releasing of 8 varieties released by forage crops and swirl millet and wheat. So without delaying much time, so I will hand over this platform to you, sir. Please take your time. Today morning, there are 2 sessions are there. Sir, I request you to finish. We will be continuing the session in the next. Sir, thank you, sir.
What time is allotted to me, sir? 10:30 to...
Sir, 11:30 you can finish.
Okay.
And then 15 minutes.
Thank you. Thank you, Dr. Raja, and all the organizers, and my friend Dr. Rajiv. And I am seeing a lot of enthusiastic people who have the lot of interest in seed production activities and how we can take up the seed production as a entrepreneur for the young generations people. So that is very the big opportunity for them to start the seed production as an entrepreneurship in their villages or in their native places, and they are having the good resources available with the public sector, as well as they can have the link up with the private sector to start this one. So today in this lecture, I have been allotted this topic, seed production, quality control, and so can you see the screen? Hello.
Yes, sir. We can see the screen.
Okay.
But it is not in full screen mode. Yes, now.
Yes, it is seed production, quality control, and value addition. This is the topic given to me in today's lecture. So all of you might be knowing the importance of the seed. It is a very basic input for the agriculture. Whatever inputs we are utilizing in agricultural crop production, all those inputs are they are very much dependent on the basic input, that is seed, because it is on the seed, what quality of seed the farmer is using, the other inputs they will have their importance in the crop production. So that is the one of the most important input, and it is comprising all forms of propagules, whether we are going using any seed or any vegetatively propagated material that forms the seed. And it is known that the improved varieties and habits which are having the good quality of seeds, they are contributing about 45% to the total productivity, and the quality seed alone can be giving the contribution up to 10 to 20%. So that is the importance of the using the quality seed, and it is therefore very essential that we should produce the quality seed of improved hybrids and improved varieties and make available the quality seeds to the farmers. And the most important thing, that we should supply the seed to the farmers at a right place, at a reasonable price, and at a particular time of sowing. So if the farmer is having the availability of the quality seed within its native place, and it will be very beneficial for the farmer. And if it is available, it is made available to the farmer within time, within the sowing season. If it is a kharif crop, then before the starts of the rain, if the farmer is having the access to the quality seed, then it is a very good thing for the farmers. And also importantly, whatever seeds are distributed to the farmers, they should have the reasonable price, so that the marginal or sub-marginal farmers, they can access these seeds. So this is the importance of the quality seed and how to make available the quality seeds to the farmers. So we should develop such a network, so that we can produce the seed wherever we want the seed to be used by the farmer. The seed production should be taken in that area only, so that the farmers will be benefited directly with the seed products in their own locality. So how we can, there are the different inputs in agricultural, mostly fertilizers, the disease resistance, then we have to use the insecticides, pesticides, then whatever soil moisture irrigations, then weedicides. So whatever agronomical principles we are following in a crop production, all these inputs, they are in vain if you are not using the improved seed or quality seed. So that's why the problem with the quality seed, if it is not supplied to the farmer, whatever he will be investing in all these resources, that will be useless for the farmer, unless and until he will not get the proper variety and proper quality seed, then all these inputs will be useless to the farmer. That is the importance of the improved quality seeds. So according to the climatic diversity in India, there are a lot of regions in India, agroclimatic regions, and each state is having the different agroclimatic zones also. Also, the crop to crop requirement is different. In crop like wheat, there are varieties which are very useful for timely sown conditions. There are certain varieties which are useful for late sown conditions. In maize and in bajra, there are different varieties for high input as well as low input. In different crops, we have the varieties for irrigated conditions, and we are having the varieties for non-irrigated conditions. So it is the responsibility of the seed production agency or the public sector organization or any private sector organization to produce the seeds of these different kind of varieties which are released by the breeders to the farmers. Because in India, this much is the diversity in the agroclimatic zones, from western Himalayas till to in the Andaman and Nicobar Islands and Lakshadweep Islands. We have the diversity of the climates, and the breeders and the ICR system, the National Agriculture Research System, as well as the private sector players, they are producing different varieties for different agroclimatic zones for fulfillment of the requirements of the different farmers in those areas. And it is a Herculean task before the seed production agency to produce the quality seed of these varieties which are released by the breeders. So what principles we can follow to produce the quality seed? So we should have the proper infrastructure, like infrastructural facilities available. Infrastructure, that means the sound R&D unit should be there to develop new and new varieties of the crop and different crop varieties. Then there should be a network of different seed production agencies for production of breeder seed. So breeder seed is the sole responsibility of the original plant breeder who has developed that variety. So the breeder seed production chain should be there, and then the certified seed and foundation seed production program, that should be handled by the sound seed production technology program. And all these, if we can have the meaningful combination of all these three factors, then it will be easy for us to make available the quality seeds to the farmers. So what is the seed quality? It is there. So it is the actually the sum of all those attributes which differentiate the seed from the grain. So all of you should be knowing the difference between the seed and grain. So the seed should be different from the grain. It should have the genetic quality, it should have the physical purity and all the germination and vigor test, all the seed moisture. So it should fulfill all the requirements. And this from time immortal, we are knowing the quality, importance of the quality seed, because subijam sukshite jayate sampadayate. This is the proverb in the Sanskrit, means that good seeds on the good land yields the abundant produce. And this was the in our yogas, in our Vedas, this was mentioned in from times immortal. Again, it was first advocated by Professor Friedrich Nebo in Germany in 1869. So this is the importance of the quality seed. Why we need the quality seed? Because it is the natural delivery systems to enhance the productivity, profitability, and the resource use efficiency of the farmer. It is the best way of transfer of new technology. If we are developing a new variety to, and if we are making the seed available to these varieties to the farmer, that is the best transfer of new technology. So seed is the carrier of new transfer of new technology, and we have experienced this transfer, the power of the seed as a transfer of new technology, because in Green Revolution of India, the seed was the best input which has transformed our Indian agricultural system, and we got the fruits of the Green Revolution due to the improved seeds available. They were made available to the farmers in Punjab and adjoining states in a very rapid manner. That's why we get the very increase in the productivity of the wheat. So that has to be continued. We have to supply the good quality seeds. So these are the importance of this good quality seeds. The basic is the crop establishment. If there is a requirement of number of plants or number of plant populations, we have to maintain in a hectare, so that we can get the optimum yield out of the crop production area. So that is the crop establishment is the first objective which we can serve with a good quality seed. Then also uniformity and purity. So elimination of the weeds, pests and diseases. These are the certain importance characters of the quality seeds, and these are the parameters through which we can assess the quality of the seeds: high genetic purity, physical purity, high germination, high vigor, and it should be free from seed-borne diseases. And the latest importance is a factor which is giving the quality seed, the what value addition we can add to the quality seed. So suppose we got the very good quality of the genetic purity seed, but how we can improve that quality of the seed so that the farmers will be benefited. So that is the value addition we can add to the parameter as a quality seed. Then safe moisture content is of course it is very important for the quality seeds. How we can produce the quality seeds? So we have to consider two main factors for quality seed production. First, what variety we should go for seed production. It should be a very improved variety and a recommended variety for that particular region. So we should have the sound knowledge of the varietal varieties available for that particular area. What are the varieties notified for that area? Those varieties should be included in the seed production program. And what quality of seed we are producing in that, it is dependent how and where it will be grown, whether the seed grower is having the full knowledge of the genetic constitution of that particular variety, and he should have the basic principle of the quality seed production. Otherwise, it will be very difficult for the farmer or the seed production agency. If you are putting a lot of effort, but if you are not knowing the what is the constitution of the variety, and then it is very difficult to maintain the genetic purity of the seed. So that is the very good important points you should be considered while planning out any seed production program. What variety we have to engage in a seed production program? The farmer should ensure that he has to produce the quality seed of the right kind of the crop and right kind of the variety. The variety should be suitable for that particular area, and the farmer should be having the interest in developing or growing that variety in an adjoining area. That variety should be only considered for the seed production, unless and until if the farmers are not willing to accept the seed of the variety which is produced in that particular area, then it is the seed production activity will be having the less impact factor on the production on the farmers in that particular area. So the selection of the variety, it is the first point we have to consider before planning any seed production program. What is the variety? So it is the assemblage of the cultivated plants which is clearly distinguished by any characteristics when it is produced sexually or asexually, and it should retain its distinguishing character. So this is the definition of the variety, and whenever we are undertaking any seed production program, we should consider, or we should have the definition in our mind. So it should be distinguished by any other characters from the other varieties. So what characters the variety is having, the diagnostic characters, they should be different from the other varieties, and the seed production agency or the seed producer should know what characters the particular variety is having different from the other varieties, and what characters are genetically controlled, means which are when reproduce sexually reproduce any variety, what characters they will be retained in the next generation. So these two factors the seed production person should be knowing, or he should have the ample knowledge of these two things. What is the variety? What are the diagnostic characters of that variety, and how they are different from the characters of other varieties of the same crop? And also he should know what are the characters which are having the genetics behind their region, or how they are heritable from one generation to another generation. So these primary thing the seed producer should know, and he should be having the proper knowledge of these things. Unless and until he is knowing what character he has to study in seed production program, it will be very difficult for the seed production person to maintain the genetic purity of that particular variety. So this is the maintenance breeding is the what we can say is the backbone of any seed production program. Whatever seed is produced, whatever nucleus seed is to be produced, that has to be produced through the maintenance breeding program. So it is to be undertaken by the original plant breeder who is releasing that variety, and how we can do the maintenance breeding program through the nucleus seed production program. We can maintain the purity of the seed nucleus seed. So this is the just nutshell. We have to select the true to type plants, and then we have to harvest this plant, then grow the plant to row progenies or head to row progenies, and then reject the substandard rows progenies. And then after going through the table examination, you should reject the undesirable types, and whatever positive or whatever the selected rows are there, we should bulb those rows, and that will constitute your nucleus seed, and from that nucleus seed there is a generation of the breeder seed. So this is the first step towards the maintaining the genetic purity of the any seed production program. Whatever breeder seed we are procuring for undertaking any foundation program or for any certified seed production program, that breeder seed should be the progeny of the nucleus seed. So the nucleus seed production program of any crop variety is the backbone of that genetic purity standards of that variety in subsequent generation. So this is the how we can the system of seed production system is operating in our country. The nucleus seed production program, that from that we are producing the breeder seed production program as per the indents given by the different state agriculture universities to the ICR. So from that, the breeder seed foundation seed production program is conducted by the different state seed certification agencies as well as the National Seed Corporation of India. And from these foundation, we are growing the certified seed production, and at last phase where we can undertake the truthfully labeled seed. So any farmer or informal systems can be doing this breeder seed, this foundation seed and certified and truthful seed production program. But the breeder seed production program is to be undertaken by the state agriculture universities only, either through the original plant breeder or through the sponsored plant breeder. So these are the steps: nucleus seed from breeder. From nucleus we can do the breeder seed, and breeder seed can be used for foundation stage, seed stage, first stage, and then, or you can go for, from foundation stage 1 you can directly go for certified seed stage, or there is another step, that is foundation seed stage 2. So from 1 you can also grow foundation stage 2, and then you can go for certified seed production program. But from foundation to certified there should be maximum 3 generations only, and this certified seed will be distributed to the commercial farmers for commercial cultivation. So the roadmap of activities: the production system, breeder, foundation, certified. Then we have to maintain the maintenance breeding in nucleus seed production. Then we should maintain the efficient agronomic management practices for any hybrid seed production program. Then there is a requirement of isolation and land requirement. The isolation, proper isolation distance is to be maintained for seed production program. In case of self-pollinated crops, the lesser will be the isolation distance. But in case of highly cross-pollinated crop like bajra or maize, the isolation distance may vary from 1 kilometer to 1.5 kilometer. And we have to maintain this isolation distance for land for the seed production program. The land requirement should be the land which is to be selected for the any seed production program, that has to be selected. That land should have the proper access with the road facilities should be there to reach the land for the field inspection. The different breeder seed production teams as well as certified seed production teams should be should have the access to those lands through the transport facilities. Then there should be a rigorous roguing operations to be conducted in a seed production program during the field inspection to rogue out the pollen shaders or off-type plants, and then there has to be a very sound certification program to be undertaken by the state certification agencies, and that has to be backed up by the grower test and seed testing facilities. So this is the roadmap of activities for any successful seed production program. So the steps in the quality seed production: selection of the proper land, proper agronomic management, proper roguing, seed health care, all should be done. The proper care should be taken during harvesting, threshing, and winnowing to avoid the admixtures. Mechanical admixture should not be there. So you have to take the care. If you are doing the harvesting through the combine harvester, the harvester should be clean before the harvesting of each of the seed production program. Then the threshing instrument should be checked properly so that there should be no mechanical admixture while threshing and drying of the seeds. Then proper cleaning, drying, and packaging of the seed which we have produced, that should be there. The seed testing for the different tests, germination and vigor should be there before the planting, and we have to advocate some good seed treatments for the farmers for the commercial seed production or commercial growing of the seed. So how we can select the site for the seed production? The seed production should be in the same climatic area for which the variety has been developed. So this is the first principle of seed production, where we are growing that variety, that area only we should undertake the seed production program. We have to consider the wind speed and direction. That the wind speed and direction should be optimum. That should be helpful for the pollen supply throughout the seed production program. The site should not be hailstorm prone site, and the high temperature and lack of soil moisture can cause desiccation of the pollens, and due to that we can get the lower quality of the seeds. So these two factors, climatic factors, should be taken into the consideration before planning any seed production program. And the ideal and the best climate is that the dry humid means that there should be no rain. There should be rain during the growth period of the crop, but there should be moderate humidity without rains during the seed development. So this is the classical area for this. This is the Ranivenur area near Bangalore. So this is the proper area where these conditions are prevailing. That's why lot of seed production of the different vegetable crops is being carried out in that particular area. So this is the selection of site. It should be proper for any seed production program. We have to fulfill all these climatic requirements before the selection of the site or selection of areas for seed production program. Then the selection of field, what field we should be selecting for any seed production program. After selection of the site, we should select the seed field which is a well-level field. It should be free from soil-borne diseases, and it should be free from volunteer plants, as well as it should be free from the weed plants. Can anybody tell me what is the definition of the volunteer plants? What is the definition of volunteer plants? Hello, are you there? Hello. 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 Am I audible to all of you?
Yes.
So what is the definition of volunteer plan? Can anybody volunteer to answer this question? What do you mean by volunteer plants? If suppose in a particular piece of land you have taken the seed production of groundnut crop in the previous season, the variety is different. Suppose the variety is TAG 224. So some seeds of the groundnut, they might be having some dormancy, and they have remain in the piece of land. So next year, when you are growing the another seed production program of that different varieties, some dormant seeds of last season, they will appear during this present season. So these plants are called as the volunteer plants. And if the varieties are of different varieties, the seeds program are of different two varieties, then these volunteer plants, they can make a problem with the genetic purity standards of the seed production lot. So the whatever field we are choosing for any selection for seed production program, that should be free from the volunteer plants. So that is the why there is a requirement that the same seed production program is to be whatever the land should be such that the seed production program of that same variety has been undertaken previous year. So this is the volunteer plan. So we should select the field which is not having the volunteer plants. Then another approach in nowadays, in case of highly pollinated crops like perlite or maize, we can go for compact area approach. So any particular village or the community, we have to adequate the people of those areas to undertake the seed production program of only one variety. So that will be very easy for the farmers to do the different field operations in a common basis. They can do the sowing with the tractor. The same tractor or same seed drill can be used for sowing of the total seed production program. And also it will be very easy for maintaining the isolation distance as well as for maintaining the purity standards while during harvesting or during drying or during threshing of the seed production program. So this is the compact area approach is to be undertaken in a way. If you want to produce the large quantity of the seeds in from a particular crop variety, then we may advocate the farmers to undertake the compact area approach, where on a land of 50-60 hectares we can undertake the seed production program of only one variety, so that it will be also very useful for the farmers to sell the seed or to outsource the seed to any private or public sector agency. So this is the importance of compact area approach. Then the rocking operation, isolation is to be maintained. What is isolation? It is necessary to avoid the unwanted supply of the pollen grains. So it may be either the space isolation or it will be a time isolation. You can have the space, or you can adjust the dates of sowing of seed production program of two different varieties, so that the pollen supply for will not be there at the same time, and that will decrease the chances of cross pollination. So we can also adequate the sowing of the border rows of the varieties, where we are taking the seed production program from the all corners. We can grow the border rows so as to protect the seed production plot from the unwanted pollen sources. So it is dependent on the isolation distance, is dependent upon the pollen shed capacity of the male parent, the wind velocity, direction of the wind, and the pollen carrying insect population. So all these, again, the whether it is a self-pollinated crop or a cross-pollinated crops, on that basis the isolation distance, it is to be decided. Then we have to do the roguing operations. It is the selective removal of the undesirable plants from the seed crops on the basis of the visual field inspection. So the roguing is a very important operation. We have to rogue out or cull out the removal of noxious weeds, the off-type plants. We have to remove from the seed production program so as to maintain the genetic purity program. Also, we have to monitor the harvesting. The early harvest gives rise to partially filled and immature seeds that will be having the high moisture content. So it will be not useful for the good quality seeds. Again, if you are delay the harvest, then there might be a chance of that the seed will be shattering, and there will be losses in seed production program. So you have to hit the optimum harvesting time. So you should know the different signs of maturity in a particular crop species, which will indicate that the seed is having the highest physiological maturity, and you should harvest the seed production program at a highest physiological maturity stage. So there are new advances where we can know the actual maturity period of the seed production program. We will see in the later slides. Then how we can maintain the quality of the seed in a seed production program. So either the breeder seed production program is undertaken by the public sector university ICR institutes. The ICR and breeder seed production program is to be monitored by the university system of seed production program, and the foundation or certified seed production program is monitored by the different state certification agencies at the field level at the seed level. So these are the different standards from which we can maintain the seed production program. So certification of the field, the source of seed is to be monitored. The preceding crop requirement, it should be checked. The isolation distance of the particular program is to be monitored from the same species, from the other species. The inseparable other crop plants, objectionable weed plants should be rogue out in a roguing, and then we can do the processing of the particular seed lot, and that will have the proper quantity of seed lot ready for the seed testing. From that seed lot, we can sample the different primary sample or submitted sample. From the submitted sample, you can prepare the working sample, and all these samples will be tested in a seed testing laboratory for different quality tests like physical purity analysis, germination test, viability, moisture test, seed health test, and the different genetic purity test. The genetic purity test is to be conducted through the other distinguishable variety test or through the grow out test. So this is the framework from which we can maintain the seed quality during the seed production program, as well as after the seed production program. We can employ the different seed quality test. So I have taken an example of maize as a seed production for a particular crop, because I came to know that in the particular area of this university, there is some growing of seed production program of maize, and you can also undertake the seed production program of maize. How we can undertake the seed production program of maize? So we can select the site for the seed production program. The plot should be on the roadside for proper monitoring. The soil should be from sandy loam to loam soils. Drainage, it should be a well-drained soil. So, hello. Soil, sandy loam to loam soil should be there, and well drained soils should be there. Then the soil should not have the moisture stress. It should be free from the cold stress also, and the excess moisture should not be there. So these are the, I can remove this one. So the excess moisture in a waterlogging conditions also, if such conditions will be there, so that will be detrimental for the quality and economical seed production program. So we should avoid such conditions for, if you are undertaking a maize seed production program, we should avoid these conditions. Then the row spacing should be between 60 centimeters between rows, and plant to plant spacing should be 20 centimeter. And we have to give the seed treatment like Captan or imidacloprid 4 gram per kg for shoot fly resistance, and this Captan should be there for Pythium stock rot, or we have to advocate the Bavestin or Captan in 1:1 ratio for triticum leaf blight or MLB or BSLB. So we have to control the seed-borne diseases in seed production program. And the isolation distance, it is dependent on the wind velocity. For inbreds, for hybrid seed production, it is almost 600 meters, and for varieties it is 300 meters. So we have to do the thinning after 10 days of germination. We have to remove the disease and insect infected and weak plants from the seed production program, and we have to give the fertilizers in recommended dosage, the N 150 kg per hectare, PTO 5 60 to 75. So total N 10% is to be given as a basal dose, and the remaining is to be given at 4 leaf, 8 leaf stage, and at the flowering and grain filling stage. So there should be split N doses for the seed production program in maize. So that is the key for seed production program, to get the good quality seeds. And we have to keep the seed production plot weed free. We can use this atrazine as a pre-emergence weedicide, and also we can advocate 1 or 2 weeding for the seed production program. And we have to give the light irrigations depending upon the climatic conditions, and there should be the irrigation at the knee-high stage, pre-flowering stage, flowering and grain filling stage, and before the maturity for the proper seed size. So we have to follow the irrigation schedule interval for the different stages in the seed production program of maize. Then we have to take the sprays for different stem borer control, and we have to do the roguing operations. So these before flowering and after flowering roguing should be there, and the roguing should be continued in a female plant. We have to detassel the female inflorescence. So what is detasseling? So it is the removal of tassel from the female plant, female parent, to ensure the cross. So all of you knowing that the maize is a monoecious crop. So both male and female reproductive organs, they are present in a particular one crop, one plant. So if you want to produce the hybrid seed, there is no system of male sterility available in maize. So in maize, if you want to make the cross between 2 parents, so either you have to remove the tassel from the female plant. So this is what we call in emasculation in other plants. In maize, we are calling this is a detasseling. We have to remove the tassel from the female parent, so that there should be a pollen supply from the pollen parent only. That is, the male parent is the only parent which can supply the pollen grains to the female parent, and so that we can get the hybrid seed produced through the detasseling operation. So this operation should be continued for almost 10 days. No female parent plant should be there which is having the tassel in the field. So you have to watch this detasseling operation in a seed production program of maize very carefully, so that there is a proper genetic purity standard maintained in a seed production program. Then you have to do the field inspection at different stages. We have to perform the field inspections to verify different genetic purity standards, to check the incidence of the seed-borne diseases, to ensure from the purity of other crop and wheat plants. So we have to observe the field inspection. What stages you can do the field inspections? You can do the field inspection at the time of sowing. So this is to be done to educate the farmers for a certified seed production program. When the inspection during pre-flowering stage, it is to be done to educate the growers regarding the plants to be rogue out and suggest other corrective measures in the seed production program. Then you have to do the inspection during flowering stage to check out the land and isolation requirement, planting ratio, source of seed, advise the farmers to continue the roguing during the flowering stages. So what factors we are to rogue out during the field inspection? First is the op type. The plants of the same crop species or the seed crop differing in the expression should be called as the op type plants, and we should remove those op type plants. Though in a hybrid seed production, you have to remove the pollen shader plants. So the plants of B line present in the A line plant, these are called as the pollen shaders. So the growers are they should be informed about the pollen shader, and they should be advocated to remove the pollen shaders during the seed field inspection. Then volunteer plants, as we have discussed, we have to remove the volunteer plants. Also, we have to discard the inseparable other crop plants or objectionable weed plants from the field, and we have to discard the disease plants. So these are the factors, these are the plants which we have to rogue out in a seed production program. So these are the different stages what we can do the field inspections during the roguing, and you have to do the harvesting at the proper moisture stage, 18 to 20%, and removal of op types from the crop from the female only, and then you can dry the cobs in a drying shed and sorting of the cobs, and there should be a shelling percentage at the 12 to 14% moisture. So we have to repeat these operations so as to maintain the genetic purity. So this is the some of the photographs of the seed production program which we have undertaken at College of Agriculture Dhule some years ago. So this is the planting we have done in the between row to row planting distance is 75 centimeter and plant to plant distance is 20 centimeter between rows. So at the initial stages we have irrigated the crop through the sub irrigation system also, and we have utilized the sprinkler irrigation systems for giving irrigations to the newly developed plants or seedlings of the maize. We have done the sowing in 2:4 ratio, so 1:2 ratio. So we have sown the 1 row of the male and 2 rows of the female. So this is the 2 rows of female, and in between there is a 1 row of male. Since this maize crop is having the protandrous nature of pollination, means the androecium that will mature first, then the gynoecium. So to make the synchronization of flowering of the female and male parent, we have to sow the female parent first. So this is the female rows. They were sown first 7 to 8 days prior to the sowing of the male parent, so that as there should be synchronization of the flowering. So you can see that these rows of male rows, they are not having the seedling emerge from the soil, but as these female rows, they were sown earlier than the male rows, there you can see the germination of the female rows. So this is the 1:2 ratio maintained in a seed production program of maize. Then at the latter stage, you can see the difference between these. These are the female rows, this is the male row. As this is sown late, so you can see that there is a difference in the height between the male and female. But as there is a first emergence of the tassel from the male parent, so this will have the synchronization of the flowering in case of. So these male rows, they should be demarcated by the growing of certain marker crops, so that at the stages like at the flowering stage, you will not know which is the male row and which is the female row. So if you grow these marker rows crops, at the very start of the male rows, then you can demarcate the male rows and female rows very easily. So this is the technique which you can employ for a hybrid seed production of maize. So this is the detasseling operation done by the last year students of BSc Agri. So whatever female rows are there, so this is the female row. We have to remove the tassel from the female row. So you have to do this operation on a very regular basis during the flowering period, so that any female plant should not be there which is having the tassel. So this is the male row plant. So from this male rows you will get the tassel only. So this is the actual proper synchronization of the flowering. You can see those. This is the female reproductive organ on the female plant, and this is the male plant. So on the male plant row you are seeing the tassel which is in full bloom, and also in a female row you are seeing the silk which are in a receptive stage. So this is, we have achieved the proper synchronization of the flowering between the male and female plants, so that there will be the supply of the pollen grains from the male parent, and it will be on the stigma of the female parent. So there will be a cross fertilization and seed setting will be there. So you will see the hybrid seed produced on the female parent in the seed production program. So this is the bird eye view of the seed production program. So you can see this clearly. These are the two female rows, and this is the one male row. So the male row is having only tassels, while all the tassels on the female rows, they have been removed through the detasseling operation. So on the larger, and you can see these are the border rows. So we have to show the 2 to 3 border rows of the male parent on the all borders of the seed production program, so as to check the unwanted supply of the pollen grains from outside source. So this is the seed production program of the hybrid maize. So you can also undertake the seed production program of the any hybrid developed by the Indian Institute of Maize Research, or any hybrid released by the Vivekananda Institute of Maize Research, Almora. So you can generate the good receipts out of the hybrid seed production program of maize. Only you have to follow the principles of hybrid seed production of maize in different stages. You have to follow this seed production program. Any questions regarding to this seed production activity? So you can also employ these public-private partnership opportunities, or you can also search for these public-private partnership opportunities. If any company is interested to do the seed production program in your area, so you can have the link up with this private partner, and you can support the seed production program of the private partner through the public-private partnership mode. So there are certain references where this PPP mode was very much useful for collaborative germplasm development, or development of different contract farming for hybrid seed production, or for extension of the human resource development. So there are certain examples for public-private partnerships in a relay race mode model. So the private sector can do the seed production program of any variety which is being developed by the public sector university. So this was in one of the examples between the Syngenta India Private Limited and the Konkan Krishi Vidyapit Dapoli. They have made a collaborative project for the seed production program of the Sahyadri rice variety. Also the JB Pant University, for they have made the MOU with the private sector for and Sankardan 3, and about 450 tons of the seed was produced. Story of the KKV Dapoli, the MOU was signed with the Syngenta, and the hybrids was spread successfully to the different parts of the India through the seed production program. Also, there was one project in Naigaon in Nanded district in Maharashtra. So the hybrid seed cotton seed production program was done with the help of the college students from the College of Agriculture Naigaon. with the help of the ProAgro company, and the ProAgro, they have extended their resources to generate the additional revenue receipts for the college. So this was the one of the story behind the PPP mode. Then there is a very encouraging stories about the ICPH 8, the first Pigeon Pea hybrid. The MOU was signed between the MICO and the ICRISAT, and MICO has established standardized the seed production program of this ICPH 8 hybrid, which is a genetically male sterile hybrid based hybrid. And they have advocated the farmers in adjoining areas of MICO company, and they have successfully produced the hybrid seed of ICPH 8. And due to that, there was a lot of increase in the productivity of the pigeon pea crop in that particular year. So this is the these are the some of the success stories of the PPP mode through which you can do the seed production program. Whenever you have completed with the seed production program, the next task is before to check the quality of the seed. So the quality of the seed can be tested through the seed testing protocols. So there are the origin of the seed testing. It has traced back into the 19th century, and in 1869 in Saxon agronomists, they have submitted one sample for botanical identification to Professor Nobe, and he has done some initial work on the seed quality testing, and that's why he was known as the father of the seed testing. So the analysis of the seed is to be done before the seed is to be sold. That was the thought of Professor Nobe in that 18th century. So this was the origin of seed testing, and Seed Testing Association in Cambridge, UK, and this was the first executive committee of the ISTA. And nowadays, ISTA is the important organization through which different quality control protocols will develop throughout the world, and they will be supplied to the different seed testing agency for the dissemination of the seed technology or quality advances. And these are the activities which is conducted by the ISTA. So there are a lot of publications, workshops they are undertaking, and they are giving the demonstrations for the development of different seed science and technology protocols to the different seed testing labs. They have the uniformity of the seed testing results. There are two orange certificate and blue certificates are issued the whole seed lot, while the blue certificate, it will report the quality of the only submitted seed sample. So these are the different ISTA proficiency tests: germination, purity test, other proficiency test, the moisture test, vigor test, seed health test, and the GMO proficiency test that has been included recently in an ISTA protocols. So through seed testing, we can do the different testing of the seeds. We have to do the seed sampling first, then we have to do the purity analysis, germination testing, and moisture testing. So the seed sampling is the most important. A sample is defined as a small portion which is drawn from the larger population. So it provides the results which can be reliably used to predict the likely or upper and lower limits of the quality of the seed lot. So this is very important how we can draw the sample. So from the samples we have to draw, there is a protocol. So you have to confirm the seed lot size, variety, lot number, and determine the number of primary samples. So it is dependent on the size of the seed lot or dependent on the number of containers from which you are drawing the sample. And from that primary sample, you can submit the this primary sample for the submitted sample, and from the submitted sample, there will be a working sample on which the different tests are to be carried out. So these are the different instruments. Seed trials, seed dividers are being used for drawing the submitted samples, and from that we have to draw the working sample. So this is the primary sample, then composite sample, submitted sample, and the working sample. So these are the seed testing. These are the sampling in how many samples we have to draw upon the lot size. If the lot is between up to 500 kg, we have to at least draw 5 primary samples. If the lot size is up to 300 kg, we have to draw 1 primary sample for each 300 kg, but not less than 5. So we have to adhere to the primary sample standards for drawing the samples. Then we have to carry out the purity analysis. We have to separate the seed lot into different pure seed, other seed component, and inert matter. So we have to take the different purity table, workboard, purity dishes, magnifying lenses. So these are the different seed dividers which we'll be using for purity analysis. Then the moisture content is one of the important tests. The orthodox is they should be having the moisture content up to 10 to 12%. And how we can ascertain the moisture content? So you have to use this formula M2 minus M3. So M1 is the weight in grams of the container and its cover. M2 and M3 are these are the weights before drying and after drying. And with this formula, you can do the moisture content. Nowadays, there are a lot of instrument which can rapidly give you the content or seed moisture content in the seeds. So these are useful. Then you can utilize different tests like the viability seed lot. So there are different stages like pre-moistening stage, and then see the so these are the on the basis of these you can decide whether the seed is viable or not. So these are the different tests for based on the viability. You have to test the germination. So between paper method, top of the paper method, sand method. So these are the standard germination tests so that you can check the quality of the seed which you have produced. To go for any other vigor test. So vigor is the manifestation of the several characters, and it can be defined as the sum total of those properties which determine the germination of seed and vigorous growth and establishment of the seedling under a wide range of growing conditions. Because vigor is different from the germination. In germination test, we are growing the seed in a normal conditions, in an optimum conditions. So here we cannot differentiate with the high vigor seed and low vigor seed. But if you expose the seeds to the range of growing conditions, to the vagaries of different climatic conditions, then only the vigorous seed will grow properly. The less vigorous seeds, they will not be successful enough in development of the proper normal seedling. So that's why we have to go for different vigor tests. So that's why we have to do this vigor testing. It should be conducted on different way. Physical parameters of the seed quality, seedling growth, physiological, biochemical indicators are there. Then we have to we can conduct the performance test under different test conditions. So test based on the physical parameters, seed size, seed density, physical soundness, or seed color. So on that basis, you can decide whether the seed is having high vigor or low vigor, and also you can check the speed of germination. So this can be determined by the simply recording the first and final count during the standard germination test, or by recording the number of seeds germinated on each day of the test, and then you can work out the speed of germination by dividing the number of seedlings and divided by number of seedling developed by number of days on which the recording is taken. So you can take the percentage and then you can compute the seedling growth rate. So the whichever doing more, that is having the more vigor. So just to check the vigor of the particular seedling. So seedling length, seedling dry weight, or seedling fresh weight, these can be done. Or you can use the different stress tests also. So physiological tests like electrical conductivity, measurement of respiratory activities like gas exchanges, production and release of volatile aldehydes, So these can be done to check the vigor. So these are the ISTA recommended methods for P. And then we can use this accelerated aging test, controlled iteration test, brick gravel test, paper piercing test, cold test, methanol stress test, and combined stress test. So all these tests will give you the bigger status of the any seed lot, whether it will be growing, if you are growing that seed lot in a particular climatic conditions, then how the seed lot will perform, that will be indicated by the vigor test. And nowadays it is very useful because the farmers, they are coming across different vagaries of nature. Suppose there is a rainfall and the farmer have sown the for the commercial cultivation, and if there is will be the seeds which can develop into the normal until you have checked the proper vigor of the seed lot, you should not discomplete. So you should develop certain specific vigor tests for specific crops based on the your research. You can develop such vigor tests so that you can give the complete germination and vigor test. So one test we can give the vigor of this particular seed lot. Then again, there is one test that is nowadays chlorophyll fluorescence test is there. So chlorophyll fluorescence test will give you the idea about the viability of the seed lot. So the maturity is directly proportional to the seed viability, and the chlorophyll presence in the is inversely proportional to the maturity of the seeds. So if you are able to detect the chlorophyll content of the seed lot, then you can say that that particular seed is viable or not. So this there are different techniques nowadays available through which you can test the chlorophyll fluorescence through the different gadgets, and you can point in my lecture, that is a value addition, how you can add the value to the seeds. You can add the value to the seeds through the seed enhancement. So the seed enhancement is a range of different treatments that can improve the performance of the seed after harvesting and conditioning and before they are sown. So it includes the priming, the hardening, pre-germination, pillating, interesting, then film coating, and other treatments to control the seed-borne pathogens. So this is the comprehensive definition of seed enhancement. But the post-harvest treatments that improve the germination or seedling or facilitate the delivery of seeds and other materials required at the time of sowing, this also includes the seed enhancement. So there are three general areas: enhancement, pre-sowing, hydration treatments, and coating technologies or seed conditioning. The pre-sowing hydration treatments include the non-control water uptake systems and control systems like seed priming. So these are the technologies. Physical enhancement, you can upgrade the upgrading and cleaning of the seed lot, filling, coating, pillating, and encrusting. Then you can do the disinfection to control the seed-borne diseases or viruses. And you can also improve the value of the seeds through some certain physiological enhancement treatments like hydration techniques, seed priming. And then you can employ different IP protection and microbinding facilities to check the genetic purity of the seed lots. So upgrading and cleaning can be done by size grading, by gravity separation, by buoyancy separation, needle separation, then intended cylinder separation, X-ray separation, chlorophyll grader, and grading based on the multispectral analysis of the seeds. So these are the physical enhancement. Film coating can be done by the coating can be done for binding, for coating can be done for the flowability of the seeds. Coating can be done for slow release of the chemicals, and it can be done for the branding, and also you can do the biological embedding through the film coating. So pillating also is there, interesting. So in nutshell, you can do these different enhancement treatments to increase the speed of germination of a particular seed if you are sowing that seed in a seed. So these are the different seed treatments. So either dry treatment or wet treatment. Dry treatment, you can directly give the treatment of dust as pesticides, or you can utilize the water-based and coating. So there you can use the film coating, encrusting as well as pelleting. So these are the different types of encrusting or pelleting. So the basic form of seed encrusting is the basic coat, which can increase the weight up to 2%. If you go for complete film coat, it will gain increase in 3 to 20% incrustation, 1 to 5x incrustation with increase will be there. Then you can go for mini peeling or standard peeling. So up to 100x you can increase the size of the seed or weight of the seed, and due to that there is a very comfortably the farmers can sow the seed which are of having very small size like onion seeds or different grasses seeds or different seeds of the important floricultural crops. If we can use this coating or encrusting pelleting techniques to increase the value of the seeds, then you can sell each seed to one seed. You can fix the price for one seed and you can sell the seeds as per the number instead of as per the weight. You can sell the seeds in numbers also, and the farmers will be having very easy to sow these encrusted or pelleted seeds. So this is the value addition. That is why what we have studied, we have discussed the chlorophyll fluorescence. It is also one of the technique through which you can detect the viability of the seed loss. So seed can be stored based on the chlorophyll analysis. The viable seeds, the more viable seeds, you can store the seeds up to longer duration, and the less viable seeds you can distribute into the market for the use in the at the present situation. You can use the less viable seeds, and if there are certain lots which are having the high viability on the basis of chlorophyll fluorescence, you can put these seed lots in the storage. So you can reduce the losses in seed due to the less germination. So this is how we can add value to the different seeds. So you can use the multispectral analysis for the different seeds, and you can then detect the fusarium infected seeds in case of wheat. If you do the multispectral analysis, so the multispectral analysis, whatever we are seeing in daylight, if you increase the wavelength or decrease the wavelength, so you can use this invisible spectrum, and then in this invisible spectrum, these different materials, they will reflect differently under different light conditions. So if you use this multispectral analysis, so with the naked eye you cannot detect the fusarium infected wheat seeds, but if you use the multispectral analysis, you can see the seed lots which are having the fusarium infection. So these are the advances in different diagnostic techniques or different advances in seed coating and pelleting, which can add value to the high price seeds. So the seeds like vegetables or floricultural crops, those can be useful for the value additions, and you can make the farmers available the good quality and high value added seeds, so that the farmers will have the benefit of those using those seeds to increase the productivity of any particular crop. So this is from my side. If there are any questions from you, so the questions are welcome. Hello.
Yes, sir. Thank you so much, sir. Lecture. The shares. So I'll be. Oh. So Thank you so much for giving a elaboration about this topic on seed production and valuation.
Okay, sir. Any questions?
So that was informative and it covers all. So now it goes to the participants. If you have any questions and doubt, you can ask the sir directly. I will ask everyone. If you have any questions. Any questions, dear participants? I think no more questions. Most of our students, and they were attended this very informative lectures. I think it will be very useful for the entrepreneur seeker as well as for producing this quality seed. Thank you very much, sir. There is other speaker is waiting, so we will give the time.
Thank you, sir.
Thank you very much, sir.
I must thank you all of the organizers and giving me the opportunity to share my thoughts on seed production activity, and I will be happy to know that you are conducting such courses for the benefit of students. In future, if you have such activities are there, please ask me. I will be there for you, sir. Okay, sir.
Yeah,
thank you.
Definitely, sir.
Thank
Definitely, we will keep in touch with you, sir. If in future also we'll hope we'll be able to help our students to learn more about the seed production and quality. Thank you very much, sir.
Thank you, sir. Thank you.
I request the next speaker, Dr. Sanjay Kumar Singh. Sir, are you there?
Good morning.
Yes, sir.
Am I audible?
Good morning. Very good morning. Yes, audible. So before going further, I request Dr. Papsila, Assistant Professor, Plant Breeding and Genetics, College of Agriculture. She will be introducing our speaker, Dr. Sanjay Kumar Singh, Principal Scientist, Indian Agriculture Research Institute, New Delhi. He will be speaking on varietal development, evaluation, and release. Dr. Papsila.
Thank you, Raza sir. Once again, good morning to you all, and good morning to Sanjay sir.
Good morning.
Sanjay sir, Dr. Sanjay Kumar Singh. Sir, he is Principal Scientist, Plant Breeding in ICR, IIWBR, Karnal. But now in 2021, he already joined IRI New Delhi, Genetics Department. And he was graduated and post-graduated with J.S. Agarwal Memorial Gold Medal from CSE University of Agriculture and Technology, Kanpur, UP, and doctorate in genetics and plant breeding from BHU Varanasi. Sir has published more than 370 in national and international journals, and he also recipient of several awards, including ICR Lal Bahadur Shastri Young Scientist Award and many other awards also. So presently he is associated with Quality Improvement Program of the IRI New Delhi. So thank you, sir, for joining with us. Now we would like to hand over the platform to Sanjay Kumar sir for his presentation. So sir, please continue for your presentations.
Good morning, dear friends and colleagues. I am sharing my slides just a minute. Can you permit for me to share? My window is not looking there.
You can share now. It is You are a co-host, now you can share.
Is it visible? Hello.
Yes, sir, it is visible now.
So good morning, dear participants and organizers. I am Dr. S.K. Singh, working as a principal scientist at IRI New Delhi. So my topic is varietal development, evolution and release. Basically, it is component of plant breeding. So I will touch in basic breedings because I understand that most of the participants are students. So maybe I can give some insight into the breeding procedures rather. So what is cultivar or variety? If you define cultivar, so it is made up of cultivated variety, culti plus var. So it is intentionally bred using cultivation methods by plant breedings. The germ plant which is intentionally bred for any purpose, that is known as cultivar or variety. Plant variety is a legal term coined by the International Union for the UPO that recognizes certain cultivars as official plant varieties in order to grant the creator of the cultivar legal protection. So when it is giving a legal protection, then it is called as a plant variety as per the terms from the UPO. So this practice is commonly known as plant breeders' right, means giving the legal protection to any of the variety to breeders. And the plant variety describes a legally protected cultivar in the system. So if you come for crops, basically there are three types of crops: self-pollinated, cross-pollinated, vegetable propagated. So I have spent about 22 years in wheat, rather include PhD, then it is more than 25 years. So most of the examples will be from field crops, and I will include some of the vegetables and horticultural crops as it is College of Horticulture and Forestry. So I will try in that direction also. So what is the step in varietal development program? Any of the program. So basic is the breeding a new variety, then its evaluation, then release process, and then seed production. So these are the major steps which are occurring in any of the varietal development procedure. So the foremost requirement, what is the requirement? So there should be first requirement as well-defined target-oriented objective. It should be there. So in most of the cases, it is yield enhancement, disease resistance, and quality improvement. Nowadays, climate resilience and abiotic stress tolerance is another point. And if you go for some of the specific crops like vegetables or horticultural products, the product specific product development is varietal development is another objective. In vegetables, there are a number of objectives: breeding for different maturity groups, for wider reproductivity, for photosensitivity, growth habits, fruit size, shape, etc. So if you come for methods of breeding, so in self-pollinated or autogamous plants, these are the different breeding methods: introduction, pure line selections, hybridization, followed by selections, in which pedigree, bulk, backcross are there, interspecies, multi-lines, hybrids, mutation. These are the breeding methods which probably you have dealt with during your UG and PG courses. So I will not go much detail, but little bit I will touch. So in pedigree methods, it is also known as combination breeding. It is used when you have two better parents but lacking in one or two different traits. So if you want to combine both of the traits in one genotype, then it is followed usually pedigree method, and you maintain pedigree of the crosses and selection. That's why it's called as pedigree methods. And you are combining two good parents. That's why it is called combination breeding. Bulk method is mostly used for abiotic stress tolerance breeding, where you wait for suitable target environment, that if the suitable environment will come, then you will make selection. Otherwise, you will just bulk in your breeding material. So that's why it is called as bulk method. Backcross method is usually for gene transfer from a donor parent to agronomically superior parent. If you have any superior agronomically parent which is lacking in one or two traits, then from a donor parent, gene is transferred using backcross method, and mostly it is used for biotic stress resistance breeding for different diseases and all pathogens. If you come for cross-pollinated crops, same way, here is also different methodologies: introduction, mass selection, recurrent selection strategies, single, then hybridization. Hybridization means here hybrids and composites and synthetics, then interspecific mutation, breeding, polyploidy, and some molecular techniques. So if you see the cross-pollinated and self-pollinated crops, so there are 2-3 different cultivars name are there, multi-line varieties. What is multi-line varieties? So multi-line varieties are mostly in self-pollinated crops where you mix number of isogenic lines. Isogenic lines means genotypically similar but differing at only one locus. Say for example, if you take for disease resistance, yellow rust, so you have 6-7 pathotypes for which different lines you have developed as a resistant, one for one pathotype, another for second one, another for third one, and mixing the seed of these, then you get to multi-line varieties. So it is mostly used in wheat, where MLKS 10, MLKS 11, and Bitur, these 3 varieties have been released for disease resistance in Kalyan Sona background. If you come for composites and synthetic cultivars, it is commonly in cross-pollinated crops, and composite varieties produced by mixing the seeds of several phenotypically outstanding lines, and then they are allowed to open pollinate, and the seed is bulked, then it is composite variety. And when the lines which have the parental lines are having tested for GCA, general combining variety, and then they are allowed to crossing, then mixing the seeds, it is called as synthetic varieties, as in developed in some pearl millet, cauliflowers, cabbage, beetroots, and these crops. Now, asexually propagated crops, there are mostly introduction, clonal selection, mutation breeding, polyploid breeding. If you go, mostly clonal selection is there. So what is clone? The clone is individual belonging to a single clone or identical in genotype. Phenotypic variation occurs due to environmental only, means phenotype is equal to genotype plus environment plus G into interaction, all included. Theoretically, it is immortal, but only deteriorate through virus or some such attacks. It is highly heterozygous, stable, and can be propagated generation after generation without any change. So mostly it is used in vegetatively propagated crops. So there are some of the achievements through clonal selection, in which direct selection have been made, or through hybridization, means crossing 2 parents and then selection like clonal selection. So there are a number of examples in potato, sugarcane, mango, citrus, etc. So there are some of the breeding methods or some facilities which are supposed to have supportive role, like off-season nursery facility, and it is in taking the crop in off-season where normal season is not there. So it reduces the breeding cycle. Say for example, in wheat, the normal season is Rabi season in plains, but we takes another crop in summers in higher Himalayan hills. So we are able to reduce the breeding cycle, which is almost 12 years. So it goes to 6 years, and it enhances the breeding cycle. Same as integration of molecular approaches like molecular added selections or molecular added crossbreeding, in which you use some molecular techniques to identify the desirable plants in early generations. So it is one of the supportive roles. Same way, physiological parameters for faster screening against abiotic stresses. So canopy temperature, depression, NDVI, root rates are different parameters which are mostly used for abiotic breeding development. Similarly, there are some new initiatives that shuttle breeding, speed breeding, participatory approaches. You must have heard the terms. So what is shuttle breeding means? So I am sitting in Karnal headquarter for wheat, and I want to breed for some South Indian conditions. So I will use shuttle breeding, means making crosses here and evaluation, one generation in that target environment of South India, then another in again Karnal, then again South India. So it is shuttling of the material, like you play badminton or you have seen some shuttle trains between two stations. So that's why it is called as shuttle breeding. It is this breeding methodology has been initiated by Dr. Annie Borla in Mexico first time. Then speed breeding. Speed breeding means you give some photo period and thermo period facilities in glass house or some poly houses, so that you reduce this that breeding cycle or their life cycle, and you can take more than 2 or 3 crops in a season. So it enhances the speed of the breeding methodology. That's why it's called speed breeding. There are participatory approaches, participatory plant breeding, participatory varietal selection, and participatory seed production. Participatory means when you involve farmers in your any of the programs, so it is called as participatory approaches. If plant breeding, you put some of your breeding material in farmers' field and simultaneously you ask them to selection along with you. So this practice is called participatory plant breeding. When you have fixed lines, fixed varieties, and you put number of varieties in farmers' field and ask them to give their choice about the performance. So it is participatory varietal selection. And if you go for seed production at farmers' field, it is participatory seed production. So in breeding methods, foremost input is the selection of parents. It may be germplasm lines, it may be suitable agronomic base or suitable donor parent. So mostly cultivars are pure line, hybrids, composites, and synthetics. So because it is more related to horticulture and forestry in your institute, so I will deal mostly hybrids in larger perspective. So for pure line variety, you just cross 2 parents and take F2, F1, F2, F3, F4, F5 generations. Then you go for station trial, pre-yield trial, then multi-location trial, and then come to coordinated system of varietal evaluation under ECRIPS, and then release. So this is the result of a cross between 2 or more different varieties and subsequent selection through several cycles of self-pollination. So it is pure line variety. The varieties have same genetic makeup. There is highly homozygous and homogeneous. When it comes to hybrid, hybrid is the first generation of a cross between 2 genetically diverse parents. When the right parents are selected, the hybrids may have good chances of hybrid vigor and then more yield. So why we go for hybrids? Because these show 15 to 20% of yield advantage at least, and it is a universal phenomenon in which F1 shows superiority to parents, and present in all biological systems and has been exploited commercially in many agricultural crops, especially cross-pollinated crops. So if you go for definition, it is heterozygous, it is uniform in look, phenotypically, and won't breed true if so again. So if you see, there are different chromosomal setup. So it is a hybrid, whereas in pure lines, it is same at both the chromosomes. So there are different types of hybrids: mid-parent heterosis, high parent, better parent heterosis, or commercial parent. So when you take heterosis over mid-parent, average of both the parents, it is mid-parent. Then over better parent, it is high parent or commercial. When you check varieties. So these are the formulas for working out the heterosis. Probably you have all know it, so not need to elaborate much more. So if you come for yield in components which contribute to any of the yields, so heterosis in grain, it is arise from an increase in the production of one or more components. So for example, if you go for wheat, it is number of fertile tillers per plant, number of grains per year, and weight of an individual grain. That is also known as 1000 grain weight. So these components decide together to what will be the yield of any of the genotype. So why hybrids need to be promoted? Because of their early maturity, high productivity, better keeping quality, better response, and so many features due to which hybrids are promoted. So for development of hybrid, the basic requirements is commercially viable level of heterosis. There should be a commercially profitable heterosis, and there should be a commercially viable seed production system, means cheaper system which can economize the seed production system. So for requirement of hybrid development, these are the different parameters which should be considered together in mind. So in self-pollinated crops, mostly parental line development is there. For hybrid development, it is through hand emasculation, male sterility and cell communicability. Then you hybridize the parental lines and evaluate that material for performance as hybrid vigor. So in hybrid cultivar development methods, there are two methods, 3-line system and 2-line system. 3-line systems is there, which is cytoplasmic genetic male sterility, in which A, B, and R 3 lines are used. That's why it is called as 3-line system. And when you go for only 2 lines, you need only 2 lines, then it is 2-line breeding system, 2-line system of hybrid development, using chemicals, using environmental, thermoperiod and photoperiod sensitive male sterility. And genetic male sterility and cytoplasmic male sterility also comes under 2-line breeding system, because only A and B lines, A and R lines are there, not 3 lines. So this is the two terminology which commonly used in hybrid cultivars. So what is male sterility? So it is condition in which the pollen grain is unavailable or cannot germinate and fertilize normally to produce seeds. So it is of 3 types: cytoplasmic genetic male sterility, in which male sterility is controlled by the interaction of genetic factors present in cytoplasm and nuclear, then environmental sensitive male sterility and chemically induced male sterility. These are the broader terms, and under this, genetic male sterility is there, which is governed by single recessive genes. But major disadvantages in this system is that 50% plants are rejected during seed production and mostly used in chili, okra, and tomatoes. Cytoplasmic male sterility system is governed by the cytoplasmic genes present in cytoplasm, and it is mostly applicable in crops where seed is not the commercial product, like onion, carrot, cabbage, and cauliflower. Once you develop hybrid and then produce by vegetative means. So this is cytoplasmic male sterility, and it is cytoplasmic genetic male sterility where 3-line systems are there. A line is the male sterile line, B line is the maintainer line. So A line and B line has difference of only cytoplasm, that B line has a fertile cytoplasm, whereas male sterile line, A line has male sterile cytoplasm. So for maintenance, for production, maintenance means production of A line, you need to cross with B line. It is both lines are isogenic lines differing at one locus of male sterility only. So you get A line as a resultant of this cross. So it is maintained, means seed production of A line. And for getting hybrids, it is used as female parent, and you use restorer line as male parent, in which nuclear combination is dominant, which gives ultimately you fertile genotype, which are hybrids. So for hybrid development, there is alien cytoplasm, normal cytoplasm in parental lines, and when you cross, then is heterozygous conditions and you get hybrids. It is same ABR line has been depicted through figures. So CHA systems means chemical hybrid agents. When you use chemicals for making male sterile parents, you take 2 parental lines, male and female, and you spray chemicals on female lines, so that becomes male sterile. And then rope pulling and other methods are used to pollinate, and then seeds from the female lines were harvested. That is hybrid. So it has advantages, fast and relatively easy to implement. Many combinations can be exploited together. It does not require any breeding methodology or pre-breeding, but simultaneously it has number of disadvantages, like very expensive to develop and register the chemical that is used for producing male sterility. It is highly stage dependent, means it is dependent on the stage of the crop and weather conditions. It affects very adversely at number of times. And sometimes it is non-complete. There is non-complete sterility in female due to its state's specificity, and it has some phytotoxic effects also. That's why it is not much more promoted in India. If you come for environmental sensitive male sterility, it is photoperiod sensitive for cytoplasmic male sterility, photoperiod sensitive genetic male sterility, then temperature sensitive, then photothermo both sensitive, and then micronutrient deficiency means some of the boron, like boron deficiency causes male sterility, and other elements also causes sometimes male sterility. So this is the pictorial depiction of two-line hybrid system in which only one parent and two parents are used, and this is three-line in which A and B is used and crossed with R. So it is three-line system. So this is the example of wheat where we are maintaining the A line, in which you can see in between four lines are the A line, and the nearby side lines, double rows are the B lines. And when crosses is made, the seed from between four lines are A line. And if you go for microscopic studies, it looks like this male style pollen grains. These all are transparent under iodine solution, and these are these are fertile, then it will take dark stains. So this is the pictorial depiction. Here it is shown that how to produce seed of hybrids. So in the feature top, you can see the border rows are the restorer lines, in between also one restorer line, and there are a number of seamless lines in between. So you just allow them pollinate. So the seeds from the middle three rows are harvested, which are the hybrids. So there are some of the supportive rules of the supportive mechanisms to promote the hybrid seed setting. There should be requirement of flowering time. Both parents need to flower at the same time. There should be synchrony in male and female flowering. There is supplemental pollination using some of the ropes or sticks to shed more pollen to female rows. But sometimes uralic acid application is done mostly in rice, so that the emergence of the female panicles come faster and accept the ability of the female to accept pollen from the male. It is improved. Isolation of seed production plots to protect genetic purity is there, and there are different types of isolations mechanism in which distance is maintaining distance between parents, and time of planting to it is time isolation, distance isolation, time isolations, and sometimes in between two plots there is some other varieties grown, other crop is grown, so that it is called it is work as barrier. So it is another isolation. So if you see in maize, there are pollen movement from male parent to female parent because it is hybrid. It is cross-pollinated crop, so the hybrid seed is obtained from the female parent. So for development of hybrids in cross-pollinated crops, you need to develop first inbred lines. So for inbred line development, these are the different methods: standard category or your 2-row methods, bulk methods, single L method, combination method. These are the for parental line development of male or female gene pool, female or male pools, where you develop parental male lines and parental female lines, and then you cross both the lines to get hybrids like this. In this, male rows are in sides, female rows are in between and detached cell. So pollen comes from male parent only, and the seed harvested is the hybrid seed. So this is one slide which shows the how vary the different crosses: single crosses, 3-way crosses, double crosses, top crosses, variety crosses. In this number, what are the female parents, male parents, what is seed yield, seed price. So these are the shown in this table, and I will share these slides so that you can use it for your studies in future. So if you come for hybrid seed production in commercially important vegetable crops, so heterosis in vegetables was first observed in tomato in 1908 from USA. In India, it was first reported in Chile in 1933 from IRI. First hybrid was developed in bottle gourd in 1971 at IRI. Indo-American Hybrid Seeds Company was the first to develop hybrids in tomato and capsicum, and later number of hybrids have been developed. And this is the percent share of the hybrids in different crops nowadays. So these are the suitable areas for hybrid seed production in different crops. In northern hills area, these are the crops. In northern plains, some of the crops, central India and south India. So there are specific locations for different crops for hybrid seed production, and companies move through this. And this is the mechanism of hybrid seed production in different vegetable crops. There are different mechanisms used in different crops. This is shown in this table. So in vegetables, mostly used hand emasculation and pollination, in which the anther is removed through emasculation. Emasculation means emasculation means removal of the male parent, so it is called as emasculation. So this is the after emasculation, pollen is being collected, then it is sprayed on the female parts to get pollinated. So this is the planting ratio of male and female parents in different crops, and the systems used mostly for hybrid seed production in vegetables. If you come for protected conditions, there are cages, insect-proof net houses. These are the conditions in which the seed production is being done, but the basic procedure is similar, although it is on a small scale, but basic principles is same for all the crops. Testing purity is done through grow out test and under field and control condition, electrophoresis, and through DNA fingerprinting. So these are some of the popular public sector hybrids under seed production in India. Vegetables, these are also same. So finally, the varietal development is the procedure where you do hybridization, then selection in segregating generations, make advanced bursts, go for PYTs, preliminary yield trials, then station trials at multi-location at institute level under different conditions. Say for example, in wheat, it is timely sown, late sown conditions, and rainfed condition. After that, it goes to coordinated trials for evaluation. So now comes to varietal evaluation in coordinated system. So what is AICRP? AICRP means All India Coordinated Research Projects. It is on different crops. For wheat, it is AICRP wheat and barley. So it is a distinctive feature of working of the ECRIP in which teams together, and there are number of genotype, a fixed number of genotypes, fixed designs, fixed replications, fixed plots, same agronomic practices at multi-locations and different cooperators. So it is basically done to make uniform evaluation of the genotypes or varieties which have been developed at number of locations. So it is done mostly for testing stability and performance, responsiveness to different environments. It is collective testing. Checks are fixed. Checks means the varieties against which the evaluation is being done. There is regular monitoring and evaluation, and it enhances the togetherness and comparative performance is being evaluated. So there are, in trialing system, there are 3 years testing. First year, it is initial varietal trial, which is based on the more than 1 or 2 zones or 2-3 states. So it is initial varietal trial. Then comes to second year advanced varietal trial, first year, and advanced varietal trial second year. This is zone specific. One zone comprises 2 or 3 states. So the test entries are inducted in the IVTs from the breeder side based on the station trial data, and if they are superior with some superiority over the existing varieties, and in this all the centers, breeding centers in ICR institutes or SAUs or private companies can contribute for evaluation of the material. It is free for ICR and SAUs, but there are some nominal charges from the private companies. So if you go for nomenclatures, there are number of nomenclatures mostly used. IET means initial evolution trial, or URT, uniform regional trial, IVT. So these are different nomenclatures which are used in coordinated evolution systems. So this is the methodology procedure. 1 year inter-test zoning. It is example of wheat crop. And then first year AVT and then second year AVT. There are a number of trials. In criteria of promotion, in first year it is yield, performance, disease resistance and quality. Second year also same, but third year in addition, agronomical performance is also inclusive to see the performance. So there are different steps: receipt of the seed, processing, data booklets, coding. Coding is done to avoid any of the bias at the centers. So preparation of seed packets, spell, then fill out the data, recording, monitoring, and reporting of the data. This is the layout which we supply with the trials, which are sent to different cooperative centers. This is the in left hand side you can see for AVT, and in right hand side it is for NIVTs. In AVTs there are 4 replications, but in NIVTs only 2 replications are there in latest design. So along with the trial seeds, we supply the agronomical practices which are to be strictly followed at all the locations to get uniform system. Field design is there. AVT is used with 12 rows. In RBD design, mostly we show with football drills. In NIVT, it is 6-row plot size of 6 meter length. It is in simple, that is, so it is, if there are 25 entries including checks, so it is 5 by 5 design. If 36 entries, then 6 by 6 or 49 for 7 by 7 likewise. So these are the some of the mistakes which are not allowed in the trialing, that you have different replications not in one single bed, but in split beds it is not allowed, or in some bed patches it is not allowed, and trial is being rejected. There should not be blockage. There should not be deviation from the design. For example, in this there is no block effect, so it is liable to reject. So proper design should be followed. Like this is the AVT plots and this is the NIT plots on right hand side. You can see very well managed crop is there. So there are different sowing methods. Drill sowing mostly done. There are line sowing also, manual sowing also, but these are the having some problems. So mostly drill sowing is being used. So there are border rows. In net plot size, if you have 12 rows, so first and last rows is the border row. So in most of the crops, they reject it before, they leave it before harvesting and not included in the trial, so that the uniform data from the middle rows is being taken. So labeling is very much important to get proper identification. So this is the system of labels in which you put the plot number like 1, trial name, entry name is coded, and in tag there are two times it is being written, because when you put to the threshing, then half of the down ticket is put inside the bag and outside the bag upper part is being tied, so that proper identity should be maintained. And this is the picture of one very well managed layout. It is at IRWBR Karnal in bit. Then comes to recording of ancillary data, in which we provide the ancillary data booklet. So you have to record different traits, data on different traits. This is the rust diseases. I have put this slide to show you that how rust looks. Is like spots basically found on stem. That's why it is called stem rust. Brown rust is little dense on leaves, and only on leaves. That's why it's called as brown rust or leaf rust. Striped rust gives impression of stripes on the leaves. That's why it is called as striped rust, and also known as yellow rust because it is yellow in color. So this is the mechanism of recording the data. 0 means no infection, R means resistant, moderately resistant, moderately susceptible, and susceptible reactions. And the percentage of the pustules on the leaf is recorded, and then say you say 20 S means 20% of the leaves are covered with S type of pustules. So likewise, these are the recording is being done. You can see the resistant and susceptible genotypes together planted, very clear picture. Then monitoring is being done by the team. It includes breeders, pathologists, agronomists, and one member from the coordinating headquarters. So they check the sowing time and layout, plant stand of experiment, agronomical practices. They check all these parameters, and if it is liable to reject, they can reject the whole of the trial, or if there are mixtures in some of the entries, so some of the entries are rejected, and it is the very highly powerful committee. If it rejects any of the variety or any of the trial, it cannot be included in further testing at any cost. So it is the power of the team. So it evaluates the breeding trials, agronomical trials, and all nurseries conducted. You can see the monitoring team visiting the fields. There are a lot of discussions on each of the trial, and in right hand down side you can see well-managed trial. It is from Anand. Sanosra is there is one NGO, Lok Bharti. They have very well-managed trials, so I put these pictures from there. So this is the data booklet where data is being filled up. Then it is the procedure of evaluation. And then comes to identification and release. In identification and release process, the proposals based on the performance over 3 years period, average performance, the varieties which are like to be superior over the checks, the concerned breeder makes its proposal, including package of practices. It should have superiority of the test, and it is identified the varietal identification committee, which is which takes meeting during workshops. There are workshops means annual meeting of different crops or ACRIPs. So there the IC is being conducted, and then there I think the proposal is put up, and they discuss each of the proposal and decide whichever is to be identified or whichever is to be rejected. So when variety is identified, then it goes for release process. So here I put the seed minimum 10 quintals. It is for wheat. For vegetables and other crops, it is lower quantity. This committee is being constituted by ICR. DDG Crop Science in ICR is chairman of the committee, and the respective director or project coordinator from different crops are member secretary. There are more voting members in this, where the director of research of the State Agriculture University or institute where the workshop is being done is member, agriculture commissioner or nominee, director of agriculture or state or nominee, representatives of seed companies, and 2 eminent scientists. These are the voting members, and non-voting members are mostly head of the different crop divisions who present the data before the committee. So once identified, then again the seed of the varieties being submitted to NBPGR to get acknowledgement and IC numbers. Then it included with along with the BIC proceedings to proposal and submitted to the CBRC. CBRC means Central Subcommittee on Crop Standards, Notification and Release of Varieties for Agricultural Crops. It comes under Department of Agriculture, Cooperation and Farmers Welfare. And again, the chairman is DDG Crop Science, and the Deputy Commissioner Quality Control is the member secretary of the committee. And as a members, there are joint secretary seats, director of agriculture of states, representatives of seed industry, and project directors of crops. And after release or approval by of the variety by this committee, this goes to Government of India for notification in Gazette of India. And if once notified, then you can go for seed production. So only release is not necessary. Its notification is much more important for going into seed production. So I have put 2-3 slides for farmers access to seed, because there are two systems of seed to access to seed to farmers. Informal system by their own exchange and or gift or some purchase from the local market, and one is formal system in which breeding and seed production, seed quality control, distribution, and farmers, it is being through government systems. So there are formal and informal systems, and there can be collaboration between the informal and formal systems in which crop breeding institutes and seed selections, these can be collaborated with PPB and PBS activities. Variety release and production is through diversity block, improved seed technology for seed multiplication, and for diffusion, local seed business and community seed banks are there for making linkages between these formal and informal systems. Nuclears, there are seed there are 4 types of seeds. Nucleus seed, which is grown by concerned breeder. The breeder seed, which is also grown by the breeder or developing institute. And then foundation seed or certified seeds. These are being produced by the SFCI or NSC or seed companies or some seed growers or progressive farmers. There is one point to remember, that in hybrids, the parental line are the breeder seeds and the hybrids are always foundation seeds for production. And if you see the generation system of seed multiplication, so it is nucleus seed, that is also known as variety maintenance, in which nucleus seed is stage 1 and stage 2 is there. Stage 1 means, if for example, if you see take for wheat, so it is ear to row, it is NSS1, and when you go for row to plot, then it is NSS2. And the most important thing which anybody should understand, that there is no rogging in the nucleus seed stage. There is only rejection. If the row is having some of the mixtures, it is totally rejected, or any of the plot which has mixtures is rejected. There is no rogging. Rogging allowed only from breeder seed, foundation seed, and certified seed. Breeder seed is having produced from nucleus seed, which is having certification tag of yellow color. Foundation seed is produced from the breeder seed, which has white color of tag. Then certified seed is produced from foundation seed, which has azure blue color. And another class is there through fully labeled seed, which is not certified, but it has the guarantee of the producing institute or developing institute that this seed has proper germination and proper purity. So they give their own certification, that's why it is called fully labeled seed, and it has polio green of color tag. So this is all about my presentations, and I wish to acknowledge Dean College of Horticulture and Forestry, CIU Pasighat, for giving me opportunity to interact with all of you. And I am also grateful to course coordinators, especially Dr. Babshila, who has effortlessly contacted every time to me. Thank you very much. And I will be happy to have any questions.
Yeah, thank you, Mr. for a wonderful this vital evaluation. I think Northeast has this a lot of potential for the release various keys. And then there are a number of seed companies are testing trees, and quality production here is less, so it's an immense and opportunity for the entrepreneurs and the students. So very novel and specific way of saying, sir. Thank you. Because participants,
Hello. Your voice is not very much clear. Hello.
So any questions and queries? You can
I think-
also type it in the chat box
Yeah, you can mail me also.
if you have any queries and questions. So I think I'll unmute everybody. Yeah, I think one can mute and speak. Okay, if there is no questions, now I profusely I thank you, Dr. Samar Singh, Principal Scientist at New Delhi, for sparing your valuable time to educating our students and entrepreneur seekers. And it is an opportunity for us to hear from you and means through this topic. So thank you so much. In near future, if any opportunities are there, we invite you, sir. Please speak again and clarify if any is raised by the students. Thank you very much, sir. Sir, you have unmuted. Okay, dear participants, we shall meet at 8:30 p.m. the last session. So after that, I will send a feedback. Please fill all this feedback link, and then you can send to us through Google link. So after that, you will get the certificate. Next week, you will get it. Maybe 31st, we will start the certificate. So please join today afternoon. Thank you so much.